Did you know that within your body there is a second brain capable of influencing your emotions, behaviors, and various other aspects that affect the whole body? Today, we will explore the intriguing bowel universe, investigating the discovery of bacteria and the extraordinary connection between the intestine and the brain. For a long time, the intestine was seen only as the organ responsible for the digestion and absorption of nutrients. However, how do we realize that this organ plays multiple functions far beyond digestion and has an essential role in our emotional and mental health? The 17th century marked a great revolution in medical science with the invention of the microscope. Anthony van Leeuwenhoek, a Dutch scientist, was the central figure of this transformation. Although Galileo is often credited with the initial invention of the microscope, it was Leeuwenhoek who improved this instrument, allowing the detailed observation of microscopic life forms. Using a rudimentary but powerful microscope that expanded up to 300 times, Leeuwenhoek made a revolutionary discovery in 1674, the presence of tiny organisms that would later be known as bacteria. Determining exactly what were the first organisms he observed is complex, but many believe they belonged to Bacillus or Spilum genus, notable for their unique forms and the ability to be expanded by Leeuwenhoek's instruments. These bacteria, once invisible to the naked eye, were approximately 1 to 2 micrometers in size. To contextualize, 1 micrometer is equal to 1,000th of millimeter. Imagine that a hair has about 70 to 100 micrometers thick. A typical 2 micrometers bacteria would mean that approximately 50 of them aligned would have the same thickness as a hair. Now visualize a 1 millimeter space where about 250,000 of these bacteria would fit. It is important to highlight that bacteria vary greatly in size and shape, so this calculation is just an illustration. Leeuwenhoek analyzed microorganisms from various sources, including rainwater, dental material, what we know today as a bacterial plate, and even their own secretions. His detailed observations documented the presence of numerous small organisms. The terminology he used to describe them marked the beginning of a deep investigation into the complex relationship between humans and the microbial world. This pioneering discovery of Leeuwenhoek was later complemented by the works of Robert Hooke, who further enhanced the microscope. However, it was the initial observation of bacteria that aroused a lasting interest in the study of these microscopic beings and their impact on human health. Leeuwenhoek's legacy goes beyond its time. His findings paved the way for studies that revealed the vastness and complexity of the microbial world both around us and within us. Today we recognize the crucial importance of microorganisms in our body. They are not only associated with infectious diseases, but also perform vital functions, such as assisting digestion, producing vitamins and neurotransmitters, and regulating the immune system. These previously unimaginable functions continue to be illuminated through the window to the invisible that Leeuwenhoek opened, deepening our understanding of life and the complex functioning of the human body together with these small beings. With the advancement of techniques and technologies, scientists have been able to isolate, cultivate, and study microorganisms with even more details. Researchers such as Louis Pasteur and Robert Koch solidified germ theory, demonstrating that specific diseases were caused by specific microorganisms. It is estimated that trillions of bacteria, as well as viruses, fungi, and other microorganisms, coexist in harmony with us, with a particularly high concentration in our digestive system. But how do we expand our understanding of the vast variety of microorganisms that coexist in our bodies? Imagine trying to know an entire city just watching people in the main square. This is how scientists studied the body's microorganisms, limiting themselves to a small laboratory sample, ignoring the vast microbial population present in us. Many of these inhabitants were not visible in laboratory samples. So what to do to observe all microorganisms? It was when a revolutionary technique emerged, genetic sequencing. Forming as a satellite, genetic sequencing allows us to visualize the whole city of microorganisms. Suddenly we were no longer looking at the central square anymore, we could see all the alleys and hidden houses. With this new look, we discovered an incredible diversity of microbial inhabitants in our body, many of which we had never seen before. The roots of genetic sequencing dates back to the 1970s when British biochemist Frederick Sanger developed the Sanger sequencing method, which became the gold standard to decode DNA. In 1980, Sanger was awarded the Nobel Prize for Chemistry for his significant contribution. In 2007, a major breakthrough was given with the launch of the Human Microbial Project, an ambitious effort that united scientists from around the world to map microorganisms in various parts of our body. However, the importance of intestinal microbes has been recognized since the 1970s thanks to the first genetic sequencing techniques. The intestine has always been a primordial focus of research, as it houses the most diverse and numerous population of microorganisms in our body. In 1996, Dr. Michael Gershon presented to the world a revolutionary perspective on the gut, calling him according to brain. This term refers to a complex network of neurons present in our digestive system, known as the enteric nervous system. Although this second brain does not formulate thoughts or dreams like the main brain, it has a great influence on mental health, emotions, behavior, humor and various brain functions. Interestingly, the enteric nervous system can operate autonomously, without the need for direct brain instructions. However, this does not mean that they work in isolation. There is bidirectional communication between the second brain and the main brain, mediated by neurotransmitters, with serotonin being the most notable. About 90% of the body's serotonin, a neurotransmitter often associated with mood and well-being, is produced in the intestine. Dr. Gershon's revolutionary work clarified the profound interaction between our enteric nervous system, the second brain, and the diverse community of microorganisms that inhabit our intestine, known as microbiota. The central discovery is that this community is not just there to help digestion. They are active producers of chemicals that affect our mind and body. In addition to serotonin, the intestinal microbiota produces and influences a series of other neurotransmitters. For example, gamma-aminobutyric acid, GABA, is crucial to regulating neural activity and has a soothing effect on the nervous system. Certain bacteria, such as those of the genus Lactobacillus and Bifidobacterium, are associated with dopamine production related to pleasure and reward. Some intestinal bacteria have the ability to produce acetic acid, which may have a protective effect on the brain, reducing inflammation. These are just a few examples of the complexity of the intestinal microbiota and its numerous metabolic products, which means that there is still much to be discovered about its interactions and effects. Research shows that microbiota disturbances or imbalances can have broad consequences, opening doors for investigations into how the intestine can influence neurological and psychiatric conditions, from depression to neurodegenerative diseases. It is important to remember that the connection between the intestine and the brain is not limited to Dr. Gershon's research. In 1976, renowned scientists Michael Bronstein and Joseph Goldenstein, who would later be recognized for their work in cholesterol and lipid metabolism, found that the intestine was a neurotransmitter factory. This has reinforced the idea that our mental and emotional health is intrinsically linked to the health of our gastrointestinal tract. The joint work of these scientists has established the foundations for a deeper understanding of how the gut influences our well-being, paving the way for subsequent studies on the importance of intestinal microbiota in modulating our mood, appetite, and mental health in general. Before we approach the relationship between intestinal health and mental diseases, it is essential to understand what is dysbiosis. Dysbiosis refers to an imbalance in the composition of the intestinal microbiota, where beneficial bacteria are less than potentially harmful bacteria. But what causes dysbiosis? To understand this, it is necessary to visualize the gut as an ecosystem. Each present microorganism has its favorite diet and, like us, thrives or weakens based on what it consumes. The foods we eat daily directly influence the composition of this community. For example, consuming natural foods, rich in essential micronutrients, not only nourishes our body, but also creates an environment conducive to beneficial bacteria. These bacteria have the ability to metabolize and, in some cases, synthesize micronutrients, increasing their bioavailability to our body. In addition, an active lifestyle that promotes good cellular oxygenation enhances these benefits, providing even greater support for the diversity and health of our microbiota. It is crucial to understand that although the health of our intestines is arguably a key factor, the complexity of mental and behavioral conditions is not limited to a single cause. Research shows that the health of our brain is deeply linked to the health of our, according to the brain, the gut. By seeking solutions to growing mental health problems, we cannot limit our focus only on the brain in our head. We must also delve into the complex and fascinating world of microorganisms that reside in our gut.